In Drosophila, the specification of the different segments of the body happens gradually, and the events that lead to the formation of these segments begin prior to fertilization and continue through embryonic development. The events that specify the segments of the body involve a hierarchy of genes that establish the anterior-posterior axis and divide the embryo into segments that have unique identities and polarity. The first set of genes to be expressed in the oocyte are known as the maternal effect genes, and they were given this name because they're deposited into the oocyte before fertilization. So these are expressed from maternal mRNAs, and those mRNAs eventually form protein gradients. The maternal effect genes are transcription factors and translation regulators, and they activate the expression of the first set of zygotic genes, which are the gap genes. The gap genes are also transcription factors, and they're expressed in broad regions in the embryo. These are known as gap genes because when they're mutated or deleted, the larva is missing multiple contiguous segments. The gap genes then activate the pair rule genes, which are also transcription factors. The pair rule genes divide the embryo into 7 segments. The pair rule genes then activate the segment polarity genes, and these are paracrine factors that divide the embryo into 14 segments. Together, the gap genes, the pair rule genes, and the segment polarity genes activate the homeotic genes, which serve to give each segment its specific identity. The first event in Drosophila development is the specification of the different axes of the body, and that takes place prior to fertilization. In Drosophila, the oocyte is enclosed within an egg chamber. It's one of the 16 cells that are derived from the oogonium. So the oogonium is a cell that divides 4 times and produces 16 cells. And you can't see them here, but there are 16 cells. Of these, 15 become the nurse cells, and 1 becomes the oocyte. The oocyte is located posterior relative to the nurse cells. The cytokinesis between the nurse cells and the oocyte is incomplete, so there are connections between these cells, and that allows for the transport of mRNAs along microtubules. The nurse cells and the oocyte are surrounded by follicular cells. Together, these cells make up the egg chamber. The first event that occurs is the transport of an mRNA for the protein gherkin, and that comes from the nurse cells and is transported to the oocyte. Once in the oocyte, gherkin is translated into protein, and it localizes between the nucleus and the cells that will become the posterior follicle cells of the egg chamber. These posterior follicle cells do not form part of the embryo, they're part of the egg chamber. However, they're very important because when the gherkin protein binds to receptors on these cells, the follicle cells respond by secreting proteins into the oocyte that rearrange the microtubules. The microtubules are rearranged so that all the fast-growing ends of the microtubule, which are known as the plus ends, face toward the future posterior, while all the slow-growing ends, which are the minus ends, face toward the future anterior. This arrangement is important for the localization of mRNAs that will be transported from the nurse cells. And this is because the motor proteins which move along the microtubules move either toward the plus end or toward the minus end. For example, bicoid mRNA associates with the microtubule motor protein dynein. This motor protein moves toward the minus ends of the microtubules, and since all the minus ends face the future anterior, bico will become localized to the future anterior. Here, it eventually becomes translated into protein and activates the genes that are needed to form anterior structures. In addition to bicoid, other mRNAs are also deposited from the nurse cells into the oocyte. This includes the mRNA for the protein nanus, which is important for specifying the posterior structures of the embryo, and that's not shown here at the moment. But in order for nanus to become localized to the posterior, another protein, Oscar, first has to be transported to this region. This is accomplished because Oscar associates with the motor protein kinesin 1, which moves toward the plus ends of the microtubules. The Oscar protein at the future posterior then traps the nanus mRNA in this location. The result of these events, which began with the localization of the Gherkin message, eventually result in the localization of the bicoid mRNA to the future anterior and the nanose mRNA to the future posterior. So as we saw in Drosophila, the bicoid mRNA gets localized to the anterior and the nanose mRNA to the future posterior. The bicoid and nanose mRNA are translated during ovulation, which takes place right before fertilization. Since the bicoid and the nanose proteins are not trapped, they diffuse, as you can see, from their site of synthesis. Experiments show that these proteins form a gradient, with bicoid being highest at the anterior and nanose being highest at the posterior. But observing a protein at a particular location during development is not enough to conclude that a gene is necessary and/or sufficient for the development of a particular structure. So additional experiments were carried out using the lose it and move it strategies. In the normal embryo, the segments of the body are the acorn, the head, the thorax, the abdomen, and the telson or tail. The acorn gives rise to the unsegmented portions of the head, while the telson gives rise to the unsegmented portions of the tail. If an embryo is taken from a mother which is a bicoid mutant, so it means that the mother cannot pass bicoid to the oocyte, this oocyte from the bicoid deficient mother lacks all anterior structures. So it has instead two tails that flank an abdomen. It's curious that two tails form instead of just an abdomen and a tail. This happens, as we'll see later, because the genes that are activated to form the acorn are the same as those that determine the tail. What makes a difference whether a particular segment becomes the acorn or the tail is the presence of bicoid. If bicoid is introduced to the anterior of an embryo from a bicoid deficient mother, the normal phenotype is rescued. If bicoid is injected in the center, a head forms at this location. As you may have noticed, there is no acorn in the embryo when the bicoid is injected into the center. This is because the genes that are needed to form the acorn and the telson are only expressed at the most anterior and most posterior ends of the embryo. Since these genes are not expressed in the center of the embryo, no acorn forms here. Another thing you may notice is that when bicoid is injected into the middle of the embryo, the head forms at the location of injection and it's flanked by the thorax. And these results suggest that bicoid is a morphogen. At higher bicoid concentrations, the genes that are needed to form the head become activated. At lower bicoid concentrations, the genes that are needed to form the thorax become activated. Finally, if bicoid is injected into the posterior of a normal embryo, wild-type embryo, a second acorn forms followed by a head and a thorax. And an acorn can form at the site that usually becomes a tail because the signaling pathway needed to form the acorn is the same as the one that is needed to form the telson. The localization of the bicoid mRNA to the future anterior of the embryo is dependent upon its 3' UTR. If the 3' UTR was altered, this would in turn affect the location of the bicoid mRNA and as a result the fate of the anterior segments. The 3' UTR is also important for the localization of nanose, but nanose actually ends up at the posterior by diffusion. However, its 3' UTR is what allows it to interact with the protein Oscar, which traps it at the posterior. As nanose diffuses in the embryo, it is inhibited from being translated into protein by inhibitors that also bind to its 3' UTR. So bicoid is actively transported along the microtubules to the anterior of the oocyte, whereas nanose diffuses to the posterior and becomes trapped by the Oscar protein, which like bicoid was transported along the microtubules. The bicoid and nanose mRNAs will remain dormant until right before fertilization, when hormonal changes lead to the unmasking of these mRNAs. The mRNAs are translated into proteins, and the proteins diffuse from their site of synthesis. They form a gradient so that the different nuclei will become exposed to different concentrations of these proteins. In addition to bicoid and nanos, there are other maternal effect genes that contribute to the specification of the anterior-posterior axis. These include hunchback, which is important for the specification of anterior structures, and caudal, which we have encountered in previous lectures, and is important for specifying posterior structures. Whereas the bicoid and nanos mRNAs are trapped at the anterior and the posterior of the embryo, hunchback and caudal mRNA are found throughout the entire egg, as depicted graphically in this image. So how is it that one of these proteins is involved in the specification of the anterior structures and the other in the specification of the posterior structures if their mRNAs are evenly distributed throughout the embryo? As it turns out, after fertilization happens, once the bicoid and the nanos mRNAs become translated into proteins, they're not only acting as transcription factors, but also as translation inhibitors. In the anterior, bicoid will act as a translation inhibitor of caudal. So it will prevent caudal from being translated into protein in the anterior regions of the embryo. In the posterior, nanos will act as a translation inhibitor of the hunchback protein. So we can see that these proteins are acting not only to activate the certain set of genes, but to inhibit another set of genes. So for example, bicoid acts not only by activating the genes that form the head, but also by inhibiting the genes that are involved in the formation of the posterior structures. As a result of the action of bicoid, even though the caudal mRNA is found evenly distributed throughout the embryo, the protein, as you can see, is not. So bicoid is not only acting as a transcription factor that functions as a morphogen, but also a regulator of translation. And I want to remind you that morphogens are usually secreted proteins because usually, in order to form a gradient so that different cells are exposed to different concentrations of the morphogen needs to be able to leave the cell where it's synthesized. This doesn't happen if a protein is a transcription factor, because transcription factors are not secreted proteins. So transcription factors don't normally function as morphogens. But in the Drosophila embryo, there is a syncytium where the nuclei share the same cytoplasm. So different nuclei can become exposed to different concentrations of transcription factors. Bicoid will activate the genes that form different anterior structures. But why does bicoid not activate the genes needed to form the head in the thorax? Well, since bicoid is a morphogen, it activates different genes based on its concentration. The genes that are needed to form the most anterior structures have binding sites for bicoid in their enhancers, but these binding sites have low affinity for bicoid, so they only become activated at very high bicoid concentrations. The genes that are needed to form the thorax also have binding sites for bicoid in their enhancers, but those binding sites have high affinity for bicoid, so they can become activated by lower concentrations of bicoid. One of the genes that is activated by bicoid is hunchback, and this is not only a maternal effect gene, but it's also a gap gene since it's also expressed from the embryo's own genome. Together, bicoid and hunchback are going to activate other head genes that are needed to form the acorn, the head, and the thorax. As we saw earlier, when bicoid is absent from the embryo, as shown here in part B, the embryo develops two tails, and these are shown in this image as FK. And recall that the reason a second tail forms at the anterior end in the absence of bicoid is because the same signaling pathway is activated at the most anterior and the most posterior end of the embryo. The genes activated at these extremities are known as a terminal gene group. They're important for specifying the most anterior unsegmented portion of the embryo, the acorn, and the most posterior unsegmented portion, which is the tail or telson. The pathway activated by the terminal gene group is the torso pathway. And as you can see, it is activated at both the anterior and the posterior end of the embryo. What determines whether the cells at each end will become an acorn or a telson is the presence or absence of bicoid. If bicoid is present, an acorn forms, otherwise a telson forms instead. This slide summarizes most of what we discussed in this lecture. In the anterior end of the embryo, there are a set of genes that define the anterior organizing center, whereas in the posterior, there's a different set of genes that specify the posterior. These genes act not only as transcription factors to activate the genes needed to form anterior and posterior structures respectively, but also as translation inhibitors. Finally, at the most anterior and the most posterior locations of the embryo, another set of genes, which are the same at each end, act to specify the terminal boundaries, the acorn and the telson.